Well, the next section in your book goes over the mitochondria and cellular metabolism, and it's very brief. And this is the section I did warn you that we're going to cover a lot deeper. And it starts off talking just a little bit about this mitochondria, and it talks about this unique organelle, which, once again, we're going to talk in a lot greater detail. We have this unique organelle with an inner and an outer membrane, highly folded organelle, especially if the folds are on the inside. And the main thing with the organelle is that it produces lots and lots of energy. This is your main energy producer in your cell that produces that ATP, that energy molecule. Now, mind you, we're going to find out that some energy happens outside this mitochondria in the cytoplasm here. So outside, we get some energy via just stored ATP, a little bit of ATP out here, as well as in muscles. Let's say we've got creatine, which I'm sure you're probably familiar with that word, actually specifically phosphocreatine. Should actually put it right. And also glycolysis, breaking down of sugar, starts out here, and we get some ATP generation out here. However, the majority of ATP happens inside this mitochondria, known as cellular respiration, and this is why we need O2, because O2 is important in this process. Now, that all being said, all these reactions, every single one of these that I've just named that's going to produce energy for us, requires enzymes. And actually, that's where I want to go to first. We're going to come back to this mitochondria, but first I want to talk about enzymes and why they're important for cellular respiration and once again, many activities in the cell. So what do we want to say here? First of all, we're going to look at the same slide again later on, but let's go over bullet point by bullet point here. Most enzymes are proteins. In fact, several years ago, they were said enzymes are proteins. Now we realize that in some cases, RNA can be used as an enzyme, but for 99% of the time, enzymes are proteins. Now, before I even go to the other ones, I want to state something. We're going to find out that enzymes, we're going to say, lower the activation energy of a reaction within the cell. In other words, they work as catalysts. Now, you may not even know what all that means yet, but whenever someone asks, hey, what's an enzyme? What's an enzyme do? They'll say, lower the activation energy of the cell, of a reaction in the cell. And that is absolutely 100% correct. But I need you to think even bigger picture. Because we have hundreds and thousands, if not more, actually, of different chemical reactions that have to go on in our cells all the time. All right? Whether it's making new chemical products or breaking them into smaller segments, whether we're building or breaking, and we're going to get into that in a second as well, these all involve chemical reactions. Now, these chemical reactions, quite frankly, would not happen in real time if it were not for enzymes. They would take a very, very long time for these chemical reactions to happen. So another way, and why I really need you to look at enzymes, is enzymes control the biochemical activity in a cell. All right? So what's really going to control what reactions, what chemical reactions are happening in the cell are these enzymes. Because if these enzymes weren't there, they, quite frankly, in real time, the reactions wouldn't happen. So these are the specialized proteins that are going to put the chemicals together to make the reaction go through. Now, that being said, what's going to control the enzymes? What's going to control enzyme production? Well, enzymes are proteins, and that once again is your DNA. So we talk about how DNA controls the cell. Well, one way the DNA controls the activity of the cell is making the specific proteins that code for enzymes that are going to control all the biochemical activity in a cell. Hopefully that makes sense. So enzymes are proteins, which are coded by, once again, the DNA, which are going to control all the biochemistry that's going on in the cell. So let's keep going here now on these bullet points. Enzymes are often named for the molecule they work on or the substrates. So actually it's going to make it easy as far as trying to guess. If you get the name of an enzyme, you can almost sometimes guess what it's going to do by its name. And quite frankly, most of the time, they're going to end with the suffix ASE. And we've already seen that. Enzymes are specific to what substrate they work on. That's huge. They're very specific. They have a specific job as far as what chemical reactions they're going to control. Enzymes have active sites. This term is going to come up again as well, where the substrate binds. So that's where all the action happens within the enzyme. And here's another important reaction. Enzymes are not used up in a reaction, but are instead recycled. So this enzyme can catalyze the same reaction over and over and over again. They are not used up, but it's like a machine that puts the pieces together and then pushes out the product, and then we'll grab two new pieces, substrates, put it together, push out a new product. So it's just going to keep going over and over and over again until that enzyme, that protein, is degraded. And then some enzymes are aided by non-protein molecules called coenzymes, and this is very important. Some of our vitamins are very important coenzymes. And once again, we're going to see these a little bit later. So this is kind of a quick overview of enzymes. Now let's dig into a little detail here. So there are two kinds of biochemical reactions. Ones that either release energy when they're done, or ones where it's going to take energy in order for them to proceed. So if it's going to release energy, they are called exergonic. Energy is released, and that's shown by what's called a negative delta G. So this is a release of energy, and this is called Gibbs, named after, obviously, a famous scientist named Gibbs, right? Gibbs free energy. So a negative delta G is energy is released, often in the form of heat, by the way. Not always. And endergonic is, well, energy is required for the action to proceed. So we had to put energy into the system, and that's noted by that positive delta G, that positive Gibbs free energy. So a normal chemical reaction, what we look at is we say it goes from reactants to products. So in this case, if we're going in this direction from A to B, A would be the reactants and B is the products. So let's look down here at an exergonic reaction. Now notice that this is the starting level of energy for the reactants right here. And then at the end, the energy level of the products is much lower. And this is how much energy is released, and hence this is an exergonic reaction. If we look over here, once again, here's our starting level of energy for our reactants, and up here, our products, the energy is much higher, so we have to supply this much energy to make the reaction go through. So now notice, this is a more favorable energy state down here. Right? This is a lower level energy, so this would be energetically favorable. That's what this means over here on my little note. Many energetically favorable reactions, exergonic, do not happen spontaneously because they have to pass through a high energy intermediate state, and that's what this big hump is right here. In other words, even though this is energetically favorable down here because it's a lower energy, all right, we had to pass through this high energy state. We had to put energy, a little energy into it, actually in this case a lot of energy, to get more energy out in the long run. And it's kind of like the old adage, you have to spend money to make money. So it's like you have to invest energy here in order to actually make energy and actually gain energy in the long run. Okay, and once again, that high energy intermediate state where we have to invest energy here, that's called the activation energy, to activate the reaction, which we have once again over here in the slide. So here's the same thing shown again, a little bit more precise here. Here, once again, we have our reactants in this first image right here, A and B, A plus B. We're going to bring them together, but in order to bring them together, we have to add all this energy. Here's my activation energy right here, energy of activation. So there's my financial investment, if you will. But wait, my products, their final energy is down here. So now I'm going to make money, all right? And I'm going to gain energy in the long run. But in order from A to B to go to C and D, my products over here, I have to put in all this energy, and yes, I'll get energy out. This is an uncatalyzed reaction. But what do catalysts do? Chemical catalysts help by lowering the activation energy, the energy needed to bring these two together. And so it is much easier for this reaction to progress. So as you can see, but once again, by these bullet points, the uncatalyzed reaction up here has a large activation energy, but the catalyzed reaction, and in the cells, those catalysts are enzymes, that activation energy has been lowered significantly, increasing the rate of reaction. Now that's the key here. It increases the rate of reaction. Now what we're going to find is when we go from A plus B goes to C plus D, well, just as likely we can actually go the other way as well. Now mind you, if this has a lower activation energy, I'm going to redraw this actually right here, A plus B, it's going to want to go more in this direction. But so we might have more of these. So I'm going to put this a little bit smaller area over here. But eventually we're going to reach an equilibrium where as we kind of go back and forth, AE plus B goes to C plus D, but then C plus D also go to A plus B. There's going to be an equilibrium eventually. Well, what we're going to find out is that this equilibrium does not change. It's just the rate at which we get there. So we may end up with 90% over here and only 10% over here, but it's going to be constantly in this equilibrium. Okay? And the equilibrium constant does not change. It's just, once again, the rate at which we get there. That's what catalysts, that's what enzymes do. So how do they work? Well, let's look over here. On the left, we have a substrate, a big molecule. And that big molecule is going to bind an enzyme that's specific for that molecule, for that substrate, at its active site. When they bind, they form what's called the enzyme-substrate complex. And the enzyme is then going to catalyze the reaction of breaking them apart and creating the new products over here. So this is where substrate is broken down into smaller products. And here's the enzyme just like it was down here. So now it can do the reaction all over again. Once again, it's recycled. It can do it over and over again. So this enzyme can break apart many copies of the substrate over and over again to form these products. Or that's degradation. We could do the exact opposite. What about if we're making something? We're synthesizing something. So in this case, we have the substrates, the two individual chemicals over here, these two substrates, and once again, they're going to bind in that enzyme at the active site. So we have, once again, have the enzyme-substrate complex. And when we're all done, we have our combined product and the enzyme ready to do it all over again. Now, once again, this is not in your book, and I'm not going to test this on you. I just want to introduce concepts because you will see these later on. And especially because I'm going to go a little bit deeper into glycolysis and these biochemical reactions that provide us energy for our working cells. Each step in the process, there are multiple chemical reactions, and each step requires an enzyme. And so different types of enzymes as far as categories and how they actually do their job. So let's look at these very briefly. And once again, just introducing concepts here. Oxidoreductases catalyze what's called redox reactions, which are oxidation reduction reactions. So you have what's called reductases and oxidases. And this gets a little confusing. This gets into a little bit of organic chemistry. But basically, when we reduce something, we're going more negative. So we're adding electrons. I'm going to reverse electrons. And when we're oxidizing something, okay, we are removing electrons. I'm oversimplifying once again. So over here, we have lactate and pyruvate. So we're going to find out at the end of glycolysis, we get 2 pyruvate molecules. And if we don't have enough oxygen for pyruvate to be moved into the mitochondria and to get as much energy out of as possible, we go under what's called fermentation, because we need to move this pyruvate around. Once again, I'm jumping ahead of myself a little bit, but I want you to go with me for a second. We need to get rid of this pyruvate if we don't have enough oxygen, and we have a specialized enzyme which falls under this category. And notice what happens here. We have pyruvate, which has this doubly bonded oxygen right here, this carbonyl group. What we're going to do actually is we're going to actually add what's called high potential electrons from this NADH. And it's going to add a hydrogen plus 2 electrons, and we're going to end up with this over here. I know the H's don't quite add up. We're not going to worry about that right now. But basically, we've actually reduced pyruvate to lactate. All right? Now, transferases transfer a group from one molecule to another. Okay? So, over here, we have norepinephrine and epinephrine, 2 very important neurotransmitters that we're going to learn about in the nervous system and anatomy and physiology, and they are also hormones. So they fall under a specialized group called catecholamines. So once again, I'm introducing stuff that you're going to find out about later, but if we look at norepinephrine and epinephrine, we have some sort of methyl group donor. All we're doing is transferring a methyl group from some donor to norepinephrine, and we're adding it right here. Here's a methyl group. So we're transferring it. All right? So transaminases transfer an amino group. So whenever you hear that, think NH3+. And kinases, this is an important one, little asterisk here, transfer a phosphate group. We're going to run into a whole bunch of very important kinases, and there's usually energy involved here. Hydrolases. Oh, wait a minute. Let's look at this for a minute. Hydro, think water, right? And what do we know that water does very well? Well, in hydrolysis, we break down larger molecules by adding water. Well, these are enzymes that actually do hydrolysis, that catalyze hydrolysis. So we have phosphatases that'll break off those phosphate bonds, peptidases that'll break apart peptides, lipases that'll break apart fats. So here we have a triglyceride. Hey, you've seen this before. We have that triglyceride with that glycerol original backbone here, and those fatty acids that were all connected. Here's our water. We know that water will hydrolyze this, but guess what? We're going to need this lipase. We're going to need this enzyme to catalyze that reaction that we've already seen. Right? Now, lyase catalyzed removal of groups to form double bonds, or the reverse break double bonds. So here's another case where we have, once again, removal of groups. So we have a double bond here in fumarate, and then we have this fumarase, a lyase, which is going to remove that double bond, and we're going to add the OH here and the H here, right, from water. So in a way, this is another type of a hydrolysis reaction, except we're adding water. We're not breaking apart the molecule completely. We're just breaking apart the double bond here We're breaking apart one of the double bonds, and now we have a single bond. All right? And this step right here is going to be important in the Krebs cycle, the citric acid cycle, which we'll be discussing later in this module. Isomerases catalyze intramolecular rearrangements. So this is moving stuff around in a molecule that already exists. So we have 3-phosphoglycerate, phosphoglycerate mutase. Ooh, think mutant, right? Moving things around, and we're just taking this phosphate and moving it to somewhere else on the molecule. And here it is. Ligases catalyze reaction in which a carbon-carbon single bond, carbon-sulfur single bond, carbon-oxygen single bond, or carbon-nitrogen bond is made or broken. So once again, here we have a DNA strand, and through DNA ligase, we're actually adding this OH to the end. And so we're going to see this a lot with cellular reproduction, how we form DNA strands. All right, so that was a lot of detail on different types of enzymes, and please, you do not have to know the specifics. Just kind of wanted to show you how these things work at various levels. Now you have to remember that, once again, I already said this before, these reversible reaction steps represent the steps in an enzyme catalyzed reaction. All right, so we're going to break it down again. We start off with an enzyme and a substrate, and they're separate. Step 1 is to form the enzyme-substrate complex. So now the enzyme and the substrates are now connected. Step 2, oh, here's the important part right now, is a transition state. This is where actually the enzyme is taking the substrates and converting it to what it will be, the final product. So then we have an enzyme product complex, and then finally the enzyme releases the product, and we get what we finally want right here. All right, so we have an enzyme substrate complex when they first bind, then there's a transition state while it's kind of putting everything together, and then the final product bound to the enzyme, and then the enzyme and product separate. So let's talk about that enzyme substrate just a little bit, specifically the active site. Once again, the enzyme substrate complex is at the active site. And what does that include? Pockets or clefts in the surface of the enzyme. So remember, this is typically a protein. 99.9% of the time this is a protein. So the R groups, remember those functional groups in the amino acids that make it the protein? The R groups of the active site are called catalytic groups, which makes sense because that's where they're going to grab a hold of those the substrate and bring them together and catalyze that reaction. Remember, there's specificity here. So the shape of the active site, okay, has to be complementary. Has to fit to the shape of the substrate. The enzyme attracts and holds the enzyme using weak non-covalent interactions. So when you think non-covalent interactions, think hydrogen bonds. And the conformation of the active site determines the specificity of the enzyme. So once again, it has to have a very specific shape. Once again, think of it like a little molecular machine, which is going to grab the substrate as individual pieces, put the pieces together, and push out a product. So originally when we looked at enzymes and substrate, this was called a lock and key model, where the enzyme and the substrate are designed to, the enzymes designed to fit perfectly with the substrate, made to fit exactly, like a lock and a key. The substrate being the key, the enzyme being the lock. But there's a problem with this. This model fails to take into account that proteins' conformational changes accommodate the substrate. So instead of so much of a lock and a key, which is actually a good way to think about it initially, that they have to fit perfectly, what's going to actually happen more likely is as soon as the enzyme and the substrate start to meet, the enzyme undergoes conformational changes. In other words, it starts to adjust. The machine starts to undergo adaptations, and it's going to fit the substrate perfectly. In other words, it's more of a flexible pocket that undergoes conformational changes to make that substrate fit perfectly. And then once again, this enzyme at this active site will make whatever changes to that substrate that it's designed to make to undergo that reaction. So here's a quick example of how enzymes function with specifically breaking down sugar, table sugar, sucrose.
An example of how enzymes function in the body is from the enzyme sucrase. Sucrase resides on the surface of the microvilli on the intestinal epithelial mucosal cell surfaces. This animation presents a graphical representation of the way that sucrase catalyzes the hydrolysis of the common disaccharide sucrose, which we know as table sugar, into its component monosaccharides, glucose or blood sugar, and fructose or fruit sugar. Hydrolysis is accomplished because when the sucrose molecule binds to the active site of the enzyme's configuration is changed so that the oxygen bridge between the two monosaccharides is exposed to water molecules in the solvent. This exposure permits a water molecule to actually break the bond, the oxygen bridge, and attach the components of water, an OH to one of the monosaccharides and an H to the oxygen, which is still attached to the second monosaccharide. This effectively cleaves the bond between the two monosaccharides and converts the disaccharide into two separate sugars. Once this is accomplished, the enzyme's configuration is changed back to the original shape. The two monosaccharides float away, and the site becomes available for another sucrose molecule to bind, change the enzyme's configuration, and be hydrolyzed. This action can be repeated many times until the enzyme becomes denatured, is inhibited, or just wears out.
All right, so let's talk about the specificity of the enzyme-substrate complex. Once again, for the enzyme and substrate to react, surfaces of each must be complementary. So enzymes, once again, are very specific. And once again, what's known as enzyme specificity is the ability of an enzyme to bind only one or at least a very few substrates, all right, thereby catalyzing only a single reaction. So we look at these two reactions. Here we have urea and we have methylurea, which looks nearly identical. The only addition is a methyl group, the CH3, right? We have urea, everything looks the same, but we're going to take away one of these hydrogens and instead we're going to add a methyl group. So you have urea and water, and with urease, here's the enzyme, it's able to break up urea into CO2 and ammonia. But if we just add that methyl group, urease doesn't work anymore. Boom. Doesn't work. No reaction whatsoever. It has a very high degree of specificity. So classes of enzyme specificity. Absolute specificity means that enzyme only reacts with one substrate. Group specificity. Enzyme catalyzes reactions involving any molecules with the same functional group. Okay, well, now when we think functional group, we think R group, right? Linkage specificity, enzyme catalyzes the formation or breakup of only a certain category or type of bond. Or stereochemical. Remember, when we think stereochemistry, we think 3D, right? 3D structure. Enzyme recognizes only one of two enantiomers. Remember the kind of the mirror images of each other? So enzymes are very specific. And once again, they control the biochemical activities of the cell. So whenever the cell needs something to be produced or broken down, the DNA will actually have the protein made. Remember, it goes from DNA to RNA to protein to get the job done. And then what doesn't need to be done anymore, that enzyme will be degraded. So now, how does these enzymes actually work as far as promoting a faster chemical reaction? Because remember, once again, it does not change the equilibrium. I cannot state that enough. But it makes it happen faster. It changes the rate of the reaction. That's the important thing here. Enzymes change the rate of reaction. Makes it happen in, once again, real time. Otherwise, for all our purposes, it wouldn't happen. It would happen eventually, but not by the time we need it to happen. Okay? So as the substrate interacts with the enzyme, its shape changes. So as you saw in that little animation, it changed the shape of the substrate, and its new shape is less energetically favorable. We saw that in that sucrase animation. It opened up sucrose so the water could attack it and hydrolyze it. The transition state has features of both the substrate and the product, so it's kind of this intermediate state right here. It's not quite substrate, it's not quite product. And once again, this transition state falls apart to yield the product, which then will disassociate from the enzyme. But remember, look at all these arrows. It goes both ways. See how all these arrows go both ways? So just as the transition state can push it this way, it can also push it this way as far as the rate of reaction. As far as how much product we're going to get, that's its equilibrium, right? And that has to do with which is more energetically favorable in the system at that time. So possible types of transition state changes. One might be that the enzyme puts stress on the bond, creating breakage. So once again, here we have an enzyme and a substrate. We've got the complex. Oh, look at the transition state. Literally, our enzyme machine here is bending, in this case, the substrate, putting stress on it, which then gives us our enzyme product complex and our two individual products in this case. Or the enzyme might bring two reactants into close proximity. So here we have the opposite of that animation. We're going to actually bring glucose and fructose together to make sucrose. But if you put these two in solution and we wanted to, in this case, it would be a dehydration reaction, right? A condensation reaction to bring these together. We want to lose water and make sucrose. We have to have these two ends hit each other just right and release water. Well, in this case, here's the enzyme and the substrate being both the glucose and the fructose. This one would be glucose, this would be fructose, 6-ring, 5-ring. And in this case, it's going to bring them close together where we can then dehydrate it, get rid of the water, and release our product, glucose. So I hope you're seeing that. Once again, think of these enzymes as little machines getting the job done, either bringing the substrates in and putting them together so they're bond, bringing the substrate in, one big molecule, and breaking it apart, or maybe bringing it in and just rearranging it. Also, the enzymes might modify the pH of the microenvironment, so just the environment right there at the active site, donating or accepting a hydrogen ion. So once again, there's that concept of pH revisiting us. So let's look at those same bullet points that I started with in the very beginning. We said that most enzymes are proteins. And for our purposes, just whenever you think enzymes, for right now, think proteins. For all intents and purposes, they're proteins, with very few exceptions. Enzymes are often named for the molecule that they work on or the substrates. We kind of saw that with the classifications as well. Enzymes are specific. Oh, we definitely saw that. They're specific to what substrate they work on. They have active sites. That's where all the action happens, is at the active sites where the substrate binds, and they must go through that transition state. We also saw that they're not used up in a reaction, but are instead recycled and used over and over and over again. And once again, they control the rate of the reaction, not the equilibrium. The final amounts if they were given all the amount of time in the world. And then some enzymes are aided by non-protein molecules called coenzymes. I didn't get much into that. I'm going to show you an animation in a moment giving you an example of a coenzyme. Actually, let's look at a couple animations, a couple examples to give you a little bit better idea as far as the detail. So here's the first one showing your basic biochemical pathway.
Organisms contain many different kinds of enzymes that catalyze a variety of different reactions. Many of these reactions, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, a substrate is converted into a product by the first enzyme in the pathway, and the product of the first reaction then becomes the substrate for the next reaction. The product of the first reaction then becomes the substrate for the second enzyme. The sequence of reactions continues until the final product is made. When a biochemical pathway is functioning, the initial substrate is continually converted to the final product through the series of steps in the pathway.
Okay, so hopefully you see how these enzymes all work together, and once again, it's like an assembly line here. It's like an assembly line to get that final product made. But wait a minute, what if we have enough of that final product? Well, what's going to happen? Well, either we need to get rid of these enzymes, or what will often happen is while we have the active site of the enzyme right here, we might have another site on the enzyme, a regulatory site, maybe on the other end, where the final product, if we have enough of it, will hook onto that, so the enzyme's active site changes shape and no longer works. And that's a form of negative feedback. In other words, we have enough of our final product, we need to stop production. Well, the final product itself might actually hit the, say, enzyme number 1 on the back side over here, and it'll change its active site so it stops working. So then the assembly stops. When we start running out of our final product, the final product will disassociate and the whole process starts over again. In fact, let's talk about that. That's feedback inhibition.
Many of the enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction. If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
So this feedback inhibition is actually a very important concept. As once again, we're building enough product, we don't need it anymore, so the product itself inhibits the whole process. And if you continue to study with your studies and say go on to the bachelor's program and take biochemistry, you're going to study a lot about this whole concept of allosteric inhibition. Lastly, I wanted one last little animation here on those coenzymes, often made partially from vitamins. And there's a very important one that we're going to see called NAD, NAD+. And niacin, the vitamin niacin, is actually part of that. And we're going to find that this NAD plus is very important in making energy. It's going to come up both in glycolysis and in cellular respiration with that citric acid cycle. So let's take a quick look.
Cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these oxidation reactions is used to form ATP. Oxidation can be defined as the removal of hydrogens from a molecule. Since a hydrogen consists of a proton and an electron are removed during oxidation. Whenever a molecule is oxidized, hydrogens removed, another molecule must be reduced, hydrogens added to it. During an oxidation-reduction reaction in the cell, an enzyme is involved in transferring the hydrogen, proton plus electron, to a coenzyme called NAD+. This enzyme has a binding site for both the substrate and NAD+. Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all enzyme-mediated reactions, once the reaction is complete, the products separate from the enzyme and the enzyme can be used again. NADH, a high-energy electron carrier, diffuses away and is available to donate the hydrogen to other molecules.
So here's an example once again of a coenzyme, the NAD+, and it's also an example of that oxidation reduction. I know they defined it quite a little bit differently. I defined it as a transfer of electrons. They defined it as a transfer of hydrogens, but notice that it was a hydrogen plus an electron. Actually, there are high potential electrons in this transfer. Won't get into that until we get into organic chemistry and biochemistry, but the point here being is that we are reducing NAD plus to NADH and making it a high energy molecule. So NADH is going to be a high energy molecule carrier, in many ways similar to ATP. And then we're going to take the energy from this and convert it to ATP. But the other point here is, once again, in this instance, the NAD plus acts as a coenzyme. This enzyme wouldn't be able to do its job if it didn't have some place to pass this hydrogen onto, and that passed it on to the NAD plus. So hence it's a coenzyme. It allows the enzyme to do its job. So once again, a lot more detail on enzymes than your book went over. But definitely wanted to introduce you to a few concepts because it will help more later on. Now we're going to move forward and talk more about glycolysis and cellular respiration that happens to that mitochondria.